Hello students, in this video we are going to do NCERT reading of the chapter Animal Kingdom. So let's start. We will start from basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory or reproductive systems. These features are used as the basis of animal classification and some of them are discussed here. First level of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, that is, they exhibit cellular level of organization. Some division of labor activities occur here in the cells. In coelentrates, the arrangement of cells is more complex. Here the cells performing the same function are arranged into tissues, hence is called a tissue level of organization. A still higher level of organization, that is organ level, is exhibited by members of platyhelminthes and other higher phyla, while tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, molluscs, echinoderms, and chordates, organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system. Level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in platyhelminthes has only a single opening to the outside of the body that serves as both mouth and anus, and is called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types. Open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it. Second one is closed type, in which the blood is circulated through a series of vessels of varying diameters, maybe arteries, veins, and capillaries. Symmetry. Animals can be categorized on basis of their symmetry. Sponges are mostly asymmetrical, that's, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into two identical halves, it is called radial symmetry. Coelentrates, ctenophores, and echinoderms have this kind of body plan. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in any one plane, exhibit bilateral symmetry. Figure 4.1 is of bilateral symmetry. Then next we have diploblastic and triploblastic organization. It is the third basis of classification of organisms. Animals in which the cells are arranged in two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals. Example, coelentrates. An undifferentiated layer, mesoglia, is present in between the ectoderm and endoderm. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and endoderm are called triploblastic animals. Example, platyhelminthes to chordates. Fourth basis of classification of animals is coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called coelom. Animals possessing coelom are called coelomates. Example, annelids, molluscs, arthropods, echinoderms, hemichordates, and chordates. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called pseudocoelomates. Example: Eschhelminthes. The animals in which the body cavity is absent are called acoelomates. Example: Platyhelminthes. Here, figure number A, it is of coelomate animals. Figure B is pseudocoelom and figure C is acoelomate. 4.1.5 classification is segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon is known as metamerism. Next is notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some plants. Most important line is this, that it is mesodermally derived structure. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates. Example is Porifera to Echinoderms. They all are non-chordates. Classification of animals. The broad classification of animals based on common fundamental features as mentioned in the preceding section is given below. Here is the classification of animals. Kingdom Animalia. Then we have level of organization. Three levels of organization: cellular, tissue, organ or organ system level. Then we have symmetry. Three types of symmetries are there: asymmetrical, radial or bilateral. Body cavity, in which that's called coelom. We have acoelomates and pseudocoelomates or coelomates. First, we will start phylum Porifera. Members of this phylum are commonly known as sponges. Another name of poriferans are sponges. They are generally marine and mostly asymmetrical animals. These are primitive multicellular animals. Most important point is that they are multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute porous ostia in the body wall into a central cavity spongiocele, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of wastes. So the function of this water canal system, it is most important that it helps us in food gathering, respiratory exchange, and removal of wastes. Choanocytes or collar cells line the spongiocele and the canals. Digestion is intracellular. The body is supported by a skeleton made up of spicules or spongin fibers. Sexes are not separate, hermaphrodite. That's, eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation and sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. Here are given examples of poriferans. First figure is of cycon and the second one is of euspongia and this third figure is of spongilla. They all are poriferans. Examples of poriferans include cycon, Scypha, spongilla also known as freshwater sponge, euspongia also known as bath sponge. Next phylum we will move on, that is phylum Coelenterata also known as Cnidaria. They are aquatic, mostly marine, sessile or free-swimming, radially symmetrical animals. The name Cnidaria is derived from the cnidoblasts or cnidocytes, which contain the stinging capsules or nematocysts present on the tentacles and the body. Cnidoblasts are used for anchorage, defense, and for the capture of prey. Cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening mouth on hypostome, that is on upper part is mouth. Digestion is extracellular and both intracellular. Some of the Cnidarians, example corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. Polyp, that is the former polyp, is a sessile and cylindrical form like hydra, adamsia, etc., whereas the latter is umbrella shaped and free swimming like aurelia or jellyfish. The latter here refers to medusa. Those cnidarians which exist in both forms exhibit alternation of generations, that is also called metagenesis. That is, polyps produce medusa asexually and medusa forms the polyps sexually. Example is obelia. Examples of cnidarians include Physalia, also known as Portuguese man of war, Adamsia, sea anemone, Pennatula, sea pen, Georgonia,